For a long time, DNA felt fixed. Something you inherit, something that defines how your body works, and something that doesn't really change. But then the idea of gene editing started to shift that. Not in a dramatic science fiction way at first, but in a very controlled, specific way. The question became not whether DNA could change, but whether it could be edited intentionally. CRISPR did not start as a human invention. It comes from a natural system found in bacteria. These organisms use CRISPR as a defense mechanism against viruses. They store small pieces of viral DNA and use them to recognize and cut the virus if it appears again. This is interesting because it means the system was already designed for targeting and cutting genetic material. Before CRISPR, gene editing was possible but complicated and less precise. CRISPR made the process simpler and more accurate. It works by combining a guiding sequence with a cutting enzyme. The guide directs where to go and the enzyme performs the cut. That combination is what makes it efficient. The process begins with creating a guide RNA. This sequence is designed to match a specific part of the DNA. It acts like a locator, ensuring that the system targets the correct gene. The accuracy of this step is important because it determines where the edit will happen. Once inside the cell, the guide RNA binds to the target DNA sequence. This pairing is based on complementary base matching. When the match is correct, the system positions itself at that exact location in the genome. The enzyme, often Cas9, then cuts the DNA at the targeted site. This creates a break in the DNA strand. At first, that sounds destructive, but the cut is what allows the editing process to begin. After the DNA is cut, the cell attempts to repair it. This repair process can be guided to introduce changes. The cell may insert, delete, or replace sections of DNA. This is how the sequence is modified. The edit does not come directly from CRISPR alone. It comes from how the cell repairs the break. CRISPR is valued for its precision, but it is not perfect. There can be off-target effects where unintended parts of the DNA are affected. This is why careful design and testing are important in research and medical applications. CRISPR has opened new possibilities in treating genetic diseases, studying gene function, and developing new therapies. It allows scientists to modify DNA in ways that were not previously practical. This has made it a major tool in modern biology. CRISPR does not create new DNA randomly. It uses a guided system to target specific sequences and relies on the cell's own repair mechanisms to make changes. By turning a natural bacterial defense system into a precise tool, it has changed how scientists approach genetics and disease. And once you understand how controlled the process is, it becomes easier to see why it has had such a significant impact. Thanks for watching.